Vanakkam. Welcome to this video on classics in neuroscience. We have been looking at signaling molecules or neurotransmitters, the first neurotransmitters in the brain. In this video, we will look at early evidence that was provided to suggest GABA as an inhibitory neurotransmitter in the central nervous system and early evidence that suggested serotonin as a neurotransmitter in the central nervous system. GABA was first identified in mammalian brain extracts as early as 1950, and it was suggested that this had inhibitory effects. The first evidence, the first set of evidence came from the work of Avapara, Woodenfriend and Roberts. Hayashi made the suggestion in 1954 that GABA had anticonvulsant effects, suggesting that it was an inhibitory neurotransmitter. There was considerable interest on this topic. Flory suggested that there was this inhibitory substance which he called as substance I in the brain extracts that caused strong inhibition. This substance I was later identified as GABA. So, because there was considerable interest in identification of an inhibitory neurotransmitter, remember that acetylcholine had already been demonstrated as an excitatory neurotransmitter in the 1950s. So, there was a race to identify an inhibitory neurotransmitter. So, there was this conference in 1959, the interdisciplinary conference that discussed the possibility that GABA was a potential inhibitory neurotransmitter. But somehow this was met with a lot of skepticism and criticism because many of the criteria for neurotransmitters were not met. In particular, there was no evidence that was provided to suggest that GABA was indeed causing IPSP, inhibitory postsynaptic potential, because you say that it is an inhibitory neurotransmitter. In the receptors or in the receiving cell, there is an interest for us to observe an IPSP. So, if this is voltage versus time, I am observing this, this is the receiving cell or the postsynaptic cell, I am interested in observing something like that, a dip in the membrane potential or an IPSP, an inhibitory postsynaptic potential was not found. There was no clear evidence for this IPSP. In fact, there were many reports that was against this. Also, it was suggested that GABA was practically undetectable in the central nervous system of crustaceans. So, it was not clear that whether GABA was indeed a inhibitory neurotransmitter, and in fact, intracellular recordings suggested that GABA does not produce hyperpolarization. Eccles demanded that GABA must produce hyperpolarization because it is suggested as an inhibitory neurotransmitter, it must produce hyperpolarization. But it was only later realized that GABA acts by activating chloride ions and there was no need for this hyperpolarization to be produced. This was found only later. The first definitive or strong evidence to suggest GABA as an inhibitory neurotransmitter came from the group of Kravitz and his colleagues, where they found that in lobster motor nerves there was synthesis, there was evidence to suggest there was synthesis, release and action, inhibitory action in lobster motor nerves. And they also found, actually Kranjevic and Schwartz also showed that if you are applying GABA or injecting GABA, there was similar synaptic inhibition as you would find by stimulating that inhibitory neuron. I think Kravitz also provided evidence to suggest a precursor for GABA called GAD, that is glutamic acid decarboxylase, right, was a precursor to GABA that was then converted into GABA in the inhibitory neuron. By the 1970s, GABA was established as the major inhibitory neurotransmitter in various brain regions. As per current analysis, what was identified in these early studies were the fast A-type GABA sensor. Now it is understood that there are different types of GABA receptors that mediate distinct mechanisms at the postsynaptic sensors or postsynaptic sites. Now we look at evidence for serotonin as a neurotransmitter. Rapport and colleagues isolated vasoconstrictor, that is a chemical that causes constriction of the blood vessel when there is bleeding that happens, when there is clotting that is going to happen, and they called this substance as serotonin. This was then identified as 5-hydroxytryptamine or 5-HT. So we will be using serotonin and 5-HT interchangeably. So 5-HT was synthesized in 1951, and this sparked a debate or an interest in identifying the role of serotonin as a possible neurotransmitter. Remember, this was the 1950s, everybody was interested in identifying neurotransmitters. So already acetylcholine was identified, GABA was identified, but there was a lot of controversy. So a question was whether serotonin was a neurotransmitter. It was found that 5-HT was very similar or identical to a substance that was widely found in the gut and that is known to have various stimulating and depressing effects. So it was found that this was present in many different parts of the body, and there was also some evidence to connect serotonin with the hallucinogen LSD. LSD antagonizes the stimulating actions of serotonin on smooth muscles. This was found, and it was found that administration of LSD causes a significant reduction in serotonin content in the brain. It was found in 1957. In late 1950s or the 1960s, serotonin was considered as a serious candidate for neurotransmitter involving various brain functions, but only much later it was realized that serotonin was perhaps involved in mediating crucial functions like mood. So current research explores pharmacology, metabolic actions, metabolic pathways, synaptic release, serotonin transporters and brain pathways, and its significance is through its use in treating mood disorders through SSRIs, that is selective serotonin reuptake inhibitors. Whether SSRIs can be used in treatment of mood disorders, for example in treating depression, are questions that are still controversial, but there was this evidence to suggest that serotonin is a powerful neurotransmitter. With this we come to the end of this video. In this video we looked at evidence that suggested GABA as an inhibitory neurotransmitter in the central nervous system, and we looked at serotonin as a neurotransmitter in the central nervous system and how it is used in the management of mood disorders. Thank you very much for your attention.